Thank you, James. And hello, everybody. Thank you for joining us today for our Sequencing FFPE Samples Introduction webinar. So we're going to cover a few topics today. So by the end of the webinar, we hope that you understand what formalin-fixed paraffin-embedded or FFPE tissue samples are and their importance. Understand some considerations for DNA and RNA extraction from FFPE samples, and then be familiar with the Illumina library prep options for FFPE sequencing. And we'll go through both whole genome and whole transcriptome sequencing, as well as our targeted sequencing options. And then you should be able to sequence your FFPE samples. So let's move on to our introduction to FFPE samples. So FFPE is used for non-sequencing applications, such as in diagnostic and drug development, and it allows us to monitor diseases over time. And so it protects the tissues from degradation, and it maintains their cell structure and subcellular components. And this allows for the indefinite storage of the tissues as well as their nucleic acids at room temperature. And it's currently estimated that there's greater than 1 billion FFPE samples archived around the world. So the question is, how do we adapt this sample type for next-gen sequencing? So some of the challenges in utilizing FFPE in genetics come from the fact that extraction of these nucleic acids yields degraded DNA and RNA. And so this degradation and damage that occurs can lead to variability among our samples. And so many factors affect the quality of our extracted nucleic acid. And these include the time to fixation, so when that tissue lost circulation and when that formalin went through the tissue, our fixation time, so how long the tissue was in formalin, as well as the tissue thickness, the specific reagents that were used, the storage conditions of the FFPE samples, how old they are, and then importantly, what type of extraction technique did we use? We also need to keep in mind that nucleic acids from FFPE samples can have chemical modifications, one of the big ones being deamination. And so these can introduce false variant calls. So this is something that we need to be aware of. And there are things such as, careful FFPE procedures, as well as data analysis methods to overcome these modifications that can occur. So let's talk about the considerations for our nucleic acid extraction. So we have a lot of different FFPE sample types. We can have blocks, scrolls, slides, or sections. So first, we'll want to check the library prep kit documentation. Whatever kit you decide to use for sequencing, definitely consult that reference guide because there may be recommendations about how much tissue we need for the extraction. So check that out first. And so in terms of the actual kit for the extraction, there are lots of commercially available kits. A very popular one is the QIAGEN All-Prep DNA RNA FFPE kit. And there may also be options specific to the kit you're using, such as the AmpliSeqfor Illumina Direct FFPE kit. And then once our sample has been dewaxed and liquefied, the DNA is often cross-linked to itself or other proteins. This is from the formalin fixation process. And so the cross-linking makes our DNA less pure, and it doesn't perform well in PCR or sequencing-based applications. So every extraction is going to include a reverse cross-linking step. And fortunately, this can be done by heating your samples to 80 to 90 degrees Celsius for about an hour or so, depending on the protocol. And that will break up those covalent bonds that would present problems with our downstream applications. And so once we complete the extraction, we do want to assess the nucleic acid quality. And I'll go through the specifics there, but proper QC of our input is going to be key to a successful sequencing project for your FFPE samples. So let's talk about DNA quality control. This is an example bioanalyzer trace we see at the top. And I just put this here demonstrating the different, or I should say the variability between FFPE samples. We see that there's a green line there showing a more intact, high quality sample. It has a lot of high molecular weight fragments versus a more degraded FFPE sample in that light blue line, which has a broad range of fragments, including a lot of smaller fragments. And we often recommend for DNA quality control to perform qPCR. And this can be done with our Illumina FFPE QC kit. And so what this does is we're comparing the amplification potential of that FFPE DNA relative to that of a non-FFPE reference DNA. And so we get a delta CQ value calculated for each sample. And this is used to predict its performance in downstream applications. So samples that amplify at later cycles in our control DNA, those are going to be of lower quality than samples that amplify sooner and have delta CQ values closer to 0. And I also linked a bulletin here on the next couple of slides. This is our recommended quality control of FFPE samples for our Illumina FFPE supported kits. So it's a great quick reference guide that'll go through the recommended checks before we use one of the Illumina kits. So for RNA quality control, this can be done with a bioanalyzer or fragment analyzer, or even a tape station if that's what we have in the lab. And what we're doing here is we're calculating the DV200 value, or the percentage of fragments that have a length greater than 200 base pairs. And at the bottom of the slide, these are just some representative example traces. On the left, with our DV200 of 77%, we can see that most of those fragments for this particular sample are greater than 200 versus our highly degraded sample on the bottom right, where we see the DV200 of 8%. We probably don't want to be using something like this. It's not going to work well for sequencing applications. And the tech note linked here, it just goes over how this methodology of evaluating the RNA was developed. And so some of our library prep kits, and I'll get into those specifics later, do actually use the DV200 value to determine how much input we should be using for a successful library. Okay. So let's move into the largest section of the webinar today, and that's talking about our different sequencing options and the specifics of using FFPE with those. So I wanted to briefly discuss the different sequencing methods. So we have whole genome or whole transcriptome sequencing, and that's where we take everything in a sample and we randomly fragment it and add our adapters to create our library. But we also have targeted sequencing. And in this case, this is a subset of genes or regions that we're interested in. And we either use an amplicon or enrichment approach to target only what we want out of that sample. And this approach can save sequencing time. It can save on our expenses, as well as our data analysis effort. So just keep this in mind as we move through the different options. And so our two approaches for targeted sequencing, the amplicon approach is going to use primers to amplify our regions of interest. In some cases, the primer sequences are removed and then we go through a subsequent round of amplification to complete the library. as shown in the diagram here. This is in contrast to our enrichment methodology, in which case we make a whole genome library and then we use biotinylated probes to pull down only our region of interest. And so we'll look at library prep kits for each of these methods, but I'll break them down by first looking at our DNA kits, and then we'll move to RNA. And then we also have some options that are designed for both DNA and RNA. So for FFPE DNA, we can do whole genome sequencing with our Illumina DNA Prep. And for enrichment sequencing, we have Illumina DNA Prep with enrichment or TruSight DNA exome. And then amplicon sequencing, we have our AmpliSeqfor Illumina DNA panels or TruSight Tumor 15. And I'll highlight some of these kits in the coming slides. So for Illumina DNA Prep, or formerly NextEra for enrichment, while it's not officially recommended for use with FFPE samples, I recommend checking this publication outlinked here. This was a publication put out by our Illumina development scientists that demonstrates that we can, in fact, use this tagmentation-based library prep kit for FFPE samples, as long as we understand some considerations when we're working with this sample type. So they did do an input QC with qPCR, like we talked about is a very popular option for QCing FFPE DNA. And what's important to note in these figures down below, specifically, let's look at panel B, is that the yield does decrease with an increased level of degradation of our DNA sample. And this can also lead to a size shift. We actually end up shifting the average size of the library to the left or to a slightly smaller size. And this makes sense if we think about the more degraded an FFPE DNA sample is. It has a larger amount of small fragments to begin with. And so as long as we understand these considerations, we can go ahead and use Illumina DNA Prep for our whole genome sequencing of FFPE DNA. So let's talk about the enrichment-based approaches. So we do have Illumina DNA Prep with enrichment. So this is also a tagmentation-based assay. We're going to create our whole genome library, just like we did with Illumina DNA Prep. And then we use biotinylated probes to pull down our regions of interest. So we do recommend using that qPCR or the Infinium FFPE QC kit to QC your DNA input. And then we do have a full webinar linked here just discussing Illumina DNA Prep with enrichment in depth if you wanted more information about this kit specifically. This kit is compatible with several different enrichment panels. Our very popular Illumina exome panel, TruSight 1, TruSight Cancer, TruSight Cardio. And then we can also do custom DNA panels in Design Studio. And I'll talk a little bit more about Design Studio when we talk about AmpliSeq. And then if you already have an enrichment probe panel, you may be able to use it with this kit. It does support third-party panels as long as you meet the requirements in our reference guide linked here. So for our amplicon-based approaches, we have AmpliSeqfor Illumina DNA panels. And so AmpliSeq is just these highly multiplex PCR-based workflow. And so we can target a lot of regions in a single run. And we do offer that AmpliSeqfor Illumina direct FFPE DNA kit for the quick nucleic acid extraction. And for these panels, we'll want to be using 1 to 100 nanograms of DNA. Typically, we don't have a QC step here for the AmpliSeqfor Illumina DNA. So if the quality of the DNA is unknown, which it typically is with FFPE samples, we can use an input amount in the higher end of this range, but let's not exceed that 100 nanograms. We don't ever want to exceed that upper limit, but we can use more if the quality is unknown. Or you can always check the quality and use your best judgment. If these are high quality samples, we can use something in that lower range. And we have a full webinar all about the AmpliSeqfor Illumina products if you want to understand the workflow a little bit more in depth. Some of our popular DNA panels include the BRCA panel, Cancer Hotspot Panel V2. We also have a full offering of community panels. So these community panels were designed to address a broad range of diseases, and they were designed with leaders in the field. And so some of the popular panels here include our microbial or antimicrobial resistance research panels and the cardiovascular research panel. And then we can make custom designs from Design Studio. I mentioned that for Enrichment as well. Something to keep in mind for these AmpliSeq custom panels, when we select that we're working with FFPE, our amplicon length will be restricted to either 140 base pair maximum or 175 base pair maximum. We no longer have the option for the larger amplicons available when we're working with high quality DNA. And that makes sense considering that these FFPE samples contain fragmented DNA. We don't want to be trying to make too large of amplicons here. And if you want to learn more about Design Studio and making your own custom panel, we have another webinar about that as well, linked here on the slide. Our last DNA amplicon workflow option is TruSight Tumor 15. So this is going to evaluate 15 significant genes in the oncology field. Those are listed here. And for this assay, we need 20 nanograms of FFPE DNA input, and we will want to QC that with the qPCR. And if you just wanted more specific information about TruSight Tumor 15, check out that product page here. So now we'll move on to the RNA options. And so just like with our DNA, we have some whole transcriptome options. We have the enrichment sequencing options, and then amplicon sequencing options. So our first option for whole transcriptome sequencing is our newest RNA, or I should say the newest family of Illumina RNA Prep kits, is our Illumina stranded total RNA with Ribo-Zero plus. So this is going to be using an enzymatic rRNA depletion method. And we can see in the workflow diagram there at the top. So this is different from the TruSeq that I'll go through as well. And here we can use 10 to 100 nanograms of RNA from FFPE, but we need to make sure that it has a DB200 value of greater than 55%. So we want to do the RNA input QC with that bioanalyzer, fragment analyzer, or tape station trace, and make sure that we're using, these would be medium to high quality FFPE input. And of course, we have TruSeq stranded total RNA. So this is our using those biotinylated target-specific oligos and the Ribo-Zero rRNA removal beads to remove the rRNA from the sample. This is a bead-based method in contrast to the previous enzymatic method. And we have to use 100 nanograms of input here. And you'll also want to do that QC trace on the bioanalyzer. And then take a look at the reference guide. There's an appendix on pages 37 through 40. And we provide example traces showing varying levels of degraded RNA, and then how to adjust the fragmentation time based on how degraded your sample is. So once you run the input RNA QC trace, just compare those images to what we see in the reference guide here, and that will tell you how to modify the protocol. Moving on to our enrichment option. So we have Illumina RNA Prep with Enrichment. This is, again, one of our newest RNA library prep kit options. And this actually uses a pigmentation-based assay to construct the library before the enrichment portion. And so we need 20 to 100 nanograms of RNA from our FFPE. We do want to run the bioanalyzer or fragment analyzer trace, and we can use samples that have DV200 values of 36.5% or greater. And this kit works with our Illumina exome panel again, our respiratory virus oligo panel, or we have the option to create custom panels. And if that's something you're interested in, please do reach out to us in tech support, or the sales team can assist with that. Another very popular enrichment option is our TruSight RNA exome. So this kit was actually optimized for sequencing RNA from degraded samples, including those FFPE samples. And so we have that 20 to 100 nanograms of total RNA from your FFPE sample. And then this is one of the kits that uses your DV200 value, so run that quality trace. And depending on the quality of your sample, that will dictate how much input we need to use here. So for high quality samples with that DV200 of greater than 70%, we can use 20 nanograms of input. But if we have a more degraded sample with that 30 to 50% DV200, we're going to use somewhere in the range of 40 to 100 nanograms. And for samples with DV200 less than 30%, we don't recommend trying to proceed with library prep. We just don't anticipate getting a successful library out of those types of samples. So our AmpliCon options for RNA include AmpliSeqfor Illumina RNA panels. Some of the popular RNA panels include the immune response panel, our transcriptome human gene expression panel, and then again, we have custom RNA panels in Design Studio. Just like for the DNA, we don't have any recommended input QC for these panels. We want to use 1 to 100 nanograms of DNase treated total RNA. And for FFPE, we actually recommend at least 10 nanograms of input. And if we are not going to assess the quality before we go in, you can always use more input up to 100 nanograms, and that can result in a higher quality library. And keep in mind that we will need to use that AmpliSeqfor Illumina cDNA synthesis kit whenever we're using these AmpliSeqfor Illumina RNA panels. Here's just some general resources I wanted to include if you're interested in RNA-Seq. There is a 3-part webinar series that goes through all of Illumina's RNA workflows. You can probably skip part 1 if you sort of have an idea of what kit you want to use. We went through the options here today for FFPE. And then part 2 is for those best practices for our Illumina RNA Prep protocols. And then part 3 talks about our introduction to analysis. So that's a great series if you want to see an end-to-end workflow and what that involves. And then we also have a great bulletin that talks about our considerations for RNA-Seq read length and coverage. So finally, we do have some options that look at both FFPE DNA and RNA simultaneously. And these are all going to be targeted approaches, either enrichment sequencing with TruSight Oncology 500 or TruSight Oncology 170, or AmpliCon sequencing options, specifically AmpliSeqfor Illumina panels. So our TruSight oncology assay is a DNA and RNA assay that targets 523 genes. We can see in the workflow diagram on the right that the DNA and RNA libraries are actually prepared in tandem, and then they will be pooled for sequencing. And this assay is similar to TruSight Tumor 170, just that TruSight Tumor 170 is a smaller panel targeting 170 genes. And so we want to use a minimum of 40 nanograms of DNA and RNA input, and definitely check out the reference guide if we're interested in using this kit, because there are recommendations for the minimum tissue size of the tissue that we want to use for extraction here. And for our input QC, we'll use qPCR or the Illumina FFPE QC kit. And we only want to use DNA samples that have a delta CQ value of less than or equal to 5. And for our RNA samples, once we do that input RNA trace, we only want to use RNA samples that result in the DB200 of greater than or equal to 20%. So this is, just keep these QC points in mind. These are really critical for success with TSO 500 here. And we have a great webinar that goes over the library, the TruSight Oncology 500 library prep in depth. I recommend checking that out if you plan to use this library prep kit in the future. So our AmpliSeqfor Illumina DNA and RNA panels, these include the focus panel, the childhood cancer panel, the comprehensive panel V3, and then our TCR beta short read panel. So these, again, look at DNA and RNA simultaneously, but you can also use these panels for just the DNA or just the RNA if that's what you're interested in. And these have the same requirements as all of the other AmpliSeqfor Illumina panels, the 1 to 100 nanograms of DNA and RNA. And this, there's actually a great app note I recommend checking out here if you're seeing, or if you expect that your FFPE samples are of varying quality. This is a great app note that just demonstrates the performance of the focus panel with varying FFPE samples. So I summarized all of our FFPE compatible Illumina library prep kits here in the table. I didn't touch in depth about all of these, so if you do have any additional questions, definitely reach out to us in tech support or reach out to your sales team. They're very knowledgeable and they can help you choose an appropriate kit for your application as well. Some other resources that are helpful when we're trying to decide what kit we'll use for our project. We have the library prep kit selector linked here, and so you can select lots of different options you can see from these boxes here when we're on that site, including down to which sequencer we have available, and that can help us narrow down the best kit for what we want. And then if you do have some genes of interest that you wanted to target, definitely check out the gene panel finder. This is a great tool and will let you know what Illumina library prep kits are going to target your genes of interest. And so let's briefly talk about our sequencing and analysis options. On this slide here, we have all of Illumina's sequencing platforms shown left to right from lowest to highest output. When we're designing an experiment, we want to consider, what platform do we have available to us, whether that's at a core facility or a lab in our department. And then we need to consider the scope of the project. So how many FFPE samples are we planning to sequence? And then what kind of coverage do we need per sample? For a lot of these kits, we do have project recommendations right on the product page that will tell you, we're targeting 50x coverage, for example. Here's how many samples we can get on a mini-seq run. or that same information is also in our sequencing coverage calculator linked here. So if you know what kit you're going to use, definitely check out that resource, and we can play around if we have different sequencers available to see what might be the best and most cost-effective option. In terms of analysis, this is very high level that we have local and cloud-based options. So our local analysis options would be on, for example, MySeq Reporter. We have enrichment analysis options or an option for that TruSight Tumor 15 workflow. And then local run manager on the iSeq, MiniSeq, or NextSeq 550. We have DNA Amplicon apps, RNA Amplicon, enrichment workflows as well. Cloud-based options include Base-Space Sequencing Hub, where we have a lot of enrichment apps or DNA Amplicon apps. We can do cuff-linked assembly for RNA projects, and then we have our very popular Dragon enrichment app as well. And if you want more information about analysis options, I highly recommend checking out our enrichment data analysis introduction webinar, and a couple of other analysis webinars linked here. I wanted to go through I linked all of the specific recorded webinars, but we do have a whole section of them on our support site if you want to explore those. We have technical bulletins on our support site, and then a couple of FFPE specific resources. We have our FFPE sequencing and arrays education page. So just a general overview about sequencing this sample type, and then the same thing for RNA sequencing of FFPE samples. And then another app note that I didn't touch on in the slides, but it's a really good read talking about assessing the quality of DNA and RNA FFPE samples. And it pertains specifically to TruSight Tumor 170, but it's a great general assessment of assessing the quality of these samples for sequencing. And you can always reach out to us here in tech support. You can reach us by email or by phone listed here. And if you enjoyed today's webinar, I also provided registration links to our upcoming webinars in the next month. So at this time, I will take your questions.